Okay, welcome back to everyone's favorite sugar splitting pathway. We will be picking up where we left off last time with the second half of glycolysis. And once we wrap that up, we will be talking about gluconeogenesis next. To help bridge the previous lecture to the current, I think it's fitting to have our molecule of the moment be the enzyme we left off on, and that is TPI, triose phosphate isomerase. This is the fifth glycolytic enzyme, and it concludes the investment phase. I'll remind you that TPI, its job is to rapidly interconvert DHAP, dihydroxyacetone phosphate, and G3P, glyceraldehyde 3-phosphate. And TPI is one of those rare instances of a catalytically perfect enzyme, which means it's incredibly efficient, if not one of the most efficient enzymes out there. Its catalytic ratio, the kcat to km, is on the order of 10 to the 8. So this is a very fast reaction. In just a test tube, it is going to be a reversible process. However, it has a very weakly reactant favoring delta G, positive 7.5 kilojoules per mole in equilibrium conditions. However, in this cell, it almost exclusively produces the product G3P. Why is that the case? Reflect on that, and we will talk about that when we meet in class. Today's learning goals will be somewhat redundant with our previous lecture. Since we're still chugging through glycolysis, we will be illustrating the key mechanisms still of the glycolytic pathway, the emphasis being on the latter half. And as we do that, we will continue to articulate the chemical logic of these transformations. And once we wrap that up and introduce to you gluconeogenesis, we will, of course, compare the glycolytic and gluconeogenic pathways in terms of outputs and regulation. Step 6 is a turning point for glycolysis. Here we will initiate our payoff phase, which means it's time to get some energy out of this process for a change. And the enzyme that is responsible for that is G3P dehydrogenase. It will extract energy by oxidizing glyceraldehyde 3-phosphate, which is an aldehyde, and oxidizing it into 1,3-bisphosphoglycerate, which is an acid. The oxidizing agent employed here by the enzyme is NAD+. That's an empty battery. Once we go through the mechanism and oxidize this position, we will get out NADH. I want you to think of NADH as money in the bank. It may not be worth much now, but we're going to cash that in later for a bunch of ATP. Speaking of, we also, in this reaction, phosphorylate G3P for free, kind of. That's the perk of having a high energy triose like G3P, by the way. It's reactive, and because it's unstable, while we do this oxidation, we can sneak in an inorganic phosphate going from G3P to 1,3-bis-phosphoglycerates. And notice how we did that without spending any more ATP. That's why I say this phosphorylation is, for lack of a better term, for free. Yes, this reaction overall is a little bit unfavorable. Take a look at the reaction delta G, positive 6.3 kilojoules per mole. But notice how every time we have a positive delta G, the next step is going to be either super fast or super favorable, or both. Biology only had 2 billion years to perfect this. Because step 7, which is coming up, is so fast and favorable, that's going to drive equilibrium towards the product direction, because 1,3-bisphosphoglycerate will not hang around. By examining the mechanism for G3P dehydrogenase, we can appreciate how we're about to pull off this rather difficult phosphorylation without the help of ATP hydrolysis. And the hack for doing so occurs pretty early on in the mechanistic cycle. But let's start at the beginning. G3P dehydrogenase has a nuanced binding pocket with a discrete NAD plus binding domain and a catalytic cysteine residue a little bit down the ways. And take note how we've actually already deprotonated the cysteine into a thiolate. Deprotonated thiolates, are very good nucleophiles. The carbonyl carbon on G3P is going to be a good electrophile. So the first reaction will be nucleophilic attack by the thiolate of deprotonated cysteine onto that electrophilic carbonyl carbon. The product of this reaction is a functional group called a thiohemiacetal. This is a very reactive group. It is another form of tetrahedral intermediate. It is a tetrahedral intermediate where the pi system that has been moved to the oxygen as a lone pair will fall back down to the carbon that was attacked. And once it does so, we have to again break one of these 4 bonds associated with the carbon. And NAD+ will coax the breaking of the CH bond, not something that's normally easily done, and this will be effectively a hydride transfer, moving that hydrogen and its electrons onto our NAD+ coenzyme. The collapse of this tetrahedral thiohemiacetal intermediate is the redox step. Once we do the redox events, what are we left with? NADH and this thioester. This thioester is also going to be rather active. They're very prone to nucleophilic attack. And what's the nucleophile that will be attacking it? An incoming phosphate ion. So while this NADH leaves and gets replaced with NAD+, the phosphate that we've just introduced has all of these O- groups on it, which are going to be nucleophilic. That O- will attack our electrophilic thioester carbon, like so, and you can envision the steps that will occur as a result of this. We're forming a bond between the phosphate and that carbon. The pi system will be kicked up. as a lone pair onto that oxygen, then fall back down and break one of the 4 pre-existing bonds. The one that's going to go is right over there, because the thiolate, S-, is the best leaving group available. Breaking that bond will liberate 1,3-bisphosphoglycerate, and it will bring us back to where we started. NAD+ and a deprotonated cysteine ready to go at it again. Step 7 is catalyzed by phosphoglycerate kinase, and the chemical logic of this step is we finally make some ATP. We will take a super high energy diphosphotriose and coax off the phosphate in the 1 position onto an ADP molecule, giving us ATP and 3 phosphoglycerates. By the way, the phosphate we're transferring here, this is the one that we added just now in step 6. So it doesn't stay on for very long. I'll remind you that the active moving phosphates from high energy metabolites onto ADP is called substrate level phosphorylation. And this is also a good time to remind you that everything after step 4, aldolase, occurs in duplicate. So we recoup 2 ATPs at this stage. At this moment, we break even ATP-wise. Even better, we already know the mechanism for practically all kinases. It's going to be pretty similar to that of hexokinase. ADP is the nucleophile, and this phosphorus is going to be the electrophile. ADP will swoop in, attack, and break the bonds between the phosphorus and the sugar acid. Step 8, phosphoglycerate mutase takes our 3-phosphoglycerate and it will move the phosphate to the 2-carbon position, giving us 2 phosphoglycerates. The chemical logic here is that by transposing our phosphate group to the secondary alcohol, it's going to free up C3 to undergo a critical dehydration in the next step. And in more good news, I've already taught you how mutases work. Flip back to your notes in the glycogen lecture to see how phosphoryl mutases operate. In that case, it was phosphoglucomutase. This has the same mechanism as that, so no need to go over that again. The finish line is drawing closer by the minute, I promise. Step 9, enolase, will catalyze the second to last step of glycolysis. And as the name suggests, enolase will take our 2-phosphoglycerate and convert it into a phosphoenol, specifically phosphoenol pyruvates. And the way it achieves this is by eliminating and thus dehydrating the alcohol on 3C, which will prompt the rearrangement of those electrons over there to form an alkene, essentially. So the chemical logic of this step is that phosphoenol pyruvate is actually the highest energy intermediate in all of glycolysis. This this phosphoenol, is so high energy that it makes this phosphate a good leaving group. And we need that to facilitate our final step, which is our last substrate level phosphorylation. And if you think back to organic chemistry, remember the enol keto tautomerization process where enols will rapidly rearrange into a carbonyl. And so you can imagine that PEP, phosphoenol pyruvate, wants to do this tautomerization once we make it. And you can see how this would do wonders of kicking off the phosphate in the phosphoenol configuration. Enolase's mechanism is like a shortened version of step 2's phosphoglucoisomerase. That was the enzyme that featured the cis-enediol intermediate, if you recall. And we will form something similar to that here called the enolate intermediate, like so. And we can achieve this transformation through 2 successive bouts of acid-base catalysis. This reactive site lysine will partake in the first instance of acid-base catalysis. What it will do is attack and deprotonate this activated proton on the C2 position. Deprotonating that carbon-hydrogen entity will relocate the electrons of that bond right over there. This is how we go about desaturating and forming this alkene. Since we're building a bond to the carbon over here at the end, we need to break one of the bonds that were already affiliated with it. And the pi system always seems to be a good place to begin. The electrons in that pi bond will jump up onto that oxygen as a lone pair and form a discrete negative charge. And see how we have these magnesium ions? These form an oxyanion hole of sorts. This metal ion catalysis, these cofactors, will reduce the barrier towards forming this high energy enol intermediate by passivating the charge on those oxygens. Even so, this looks to me like a pretty unhappy high energy situation. So we can relieve that stress through an electronic rearrangement where one of those lone pairs on one of those oxygens will fall back down, and that's going to break the pi system that we just formed between those 2 carbons. And we will move over the alkene from those 2 carbons to the next pair downward, just like so. And when we form a bond to this carbon, we need to break one as well. The bond that ends up being broken in this second and final step is the one between carbon and oxygen of the OH. Now, OH on its own is not a good leaving group. However, that's where the second instance of acid-base catalysis comes in clutch. A proximal glutamic acid, shown over here, will protonate that OH as it leaves, forming water, H2O, and that is a good leaving group, which makes this final rearrangement possible, leaving us with phosphoenol phosphoenol pyruvate. Phosphate over here, and the double bond over there that we worked so hard to form. Congratulations, you've made it to the final step of the payoff phase, the final step of glycolysis, and that's enzyme number 10, pyruvate kinase. Its job is to transfer the phosphate group, the last phosphate group, from phosphoenol pyruvate onto an ADP molecule, yielding us pyruvate and molecule of the hour, ATP. Remember that every enzyme after step 4, aldolase, performs its reactions in duplicate because aldolase cuts its substrate in half into 2 pieces. So we're not just making an ATP molecule here, we're making 2 ATPs, bringing our total tally of ATP per glucose to 4. The net number of ATP gained, however, is only 2 ATP molecules because we lost 2 to the investment phase. Why is this important? Well, pyruvate kinase is our second and final substrate level phosphorylation event, and it is the one that induces an ATP profit. We don't make any energy technically until the very end of it. I also want you to pay attention to the reaction delta G of pyruvate kinase. It is the most negative, most exergonic step in the entire sequence, with a free energy of negative 31.4 kilojoules per mole, making it the third rate-limiting step of glycolysis. Let's briefly recap all of the regulators of glycolysis and compare them. Our first one is step 1, hexokinase. And what is that the gatekeeper of? Well, it's the gatekeeper of glucose metabolism. Its product, G6P, is very useful and very promiscuous. That substrate can hop into all sorts of metabolic pathways, glycolysis just being one of them. PFK1, phosphofructokinase, the third enzyme in the sequence, that is the gatekeeper to glycolysis itself. The third and final gatekeeper, the last step of glycolysis, pyruvate kinase. What are we gatekeeping here? The answer is aerobic respiration. The products of pyruvate kinase largely will be funneled into the aerobic respiration pathway, where we will fully dismantle and oxidize pyruvates into CO2 molecules, which will bring us a lot of ATP. However, pyruvate is also a useful molecule, and we don't want to burn any of our metabolites unless we are sure we need the energy. So that's why it's useful to tightly control the final bookend of glycolysis through allostery. Let's review the allosteric activators and the allosteric inhibitors. For the activators, we have FBP, fructose bisphosphate. This may sound familiar because it's actually the product of step 3, PFK, our second gatekeeper. Now, why is this interesting? Well, what we're effectively achieving here is communication between gatekeeper number 2 and gatekeeper number 3. If we experience a sudden need for energy and we're going to rapidly ramp up glycolysis, what PFK can do is it will create a big burst of products, some of which will diffuse over and tap PK on the shoulder and say, hey, we've got a bunch of metabolite incoming, get ready for it. So that is a pretty, I think, nifty hack that evolution has installed within the glycolytic machinery, this feed-forward stimulation. As for the downregulators, we have ATP, alanine, and acetyl-CoA. At this point, I think ATP is a little bit self-explanatory, why it's a great inhibitor for this step, because an accumulation of ATP means that we've met our energy demands. Our energy charge is rising, so stop burning fuel, please. Let's maybe use our resources for anabolism. Now, why are alanine and acetyl-CoA regulators of pyruvate kinase as well? The accumulation of either of those is a sign that we don't need the products of glycolysis anymore and we're wasting glucose. Both of these are essentially made from pyruvates. And acetyl-CoA in particular is going to be the beginning of the citric acid cycle, which is the first phase of aerobic respiration. So if we don't want to burn metabolites wantonly, we need these two to signal to glycolysis to cool it. As for the mechanism, it's a kinase, so I'm not going to go through the kinase mechanism again. I will just mention here that this reaction features a very key enol keto tautomerism, and you can get a visual of that down here. And this rearrangement is going to ultimately provide the energy needed to drive our final phosphoryl transfer. Think of it this way: the thermodynamic urge to get rid of the enol pyruvate form and make it the more stable carbonyl is what will pop off the inorganic phosphate attached over there. If you step back and take it all in, it's all nothing short of clever. Putting it all together, one round of glycolysis will consume a glucose molecule, 2 ATP molecules, and 2 NAD pluses, which will be serving as oxidants. In the process, we are going to make 2 sugar acids, 2 pyruvates, and these pyruvate molecules have many different cellular fates, which we will unpack soon enough. We will be making 4 ATP, which can be used to power the energy-dependent processes within the cell. But keep in mind, since we've spent 2 to make those 4, the net is 2. And also 2 NADH, the reduced form of NAD+. These I like to think of as money in the bank, a resource that we're going to cash in later to get a whole bunch of ATP specifically. Now, glycolysis needs to be heavily regulated. This is going to ensure that we use our resources wisely and that we only make ATP when we need it. If we don't need ATP, if the energy demands are not very high, well, we can use these resources in other pathways, like making glycogen, for example. And so if we put it all together, the entire equation for glycolysis is as follows. 1 glucose plus 2 NAD plus, 2 ADP, 2 PI yields 2 pyruvates, 2 NADH, 2 protons, and 2 ATP. It's commonly said that cancer has a sweet tooth. Before a tumor undergoes angiogenesis, which is the recruitment of blood vessels and oxygen into the tumor environment, tumor cells are rather anoxic, and they have to derive most, if not all, of their energy production from inefficient glycolysis alone. Therefore, cancer tends to be very glycolytic and therefore sucks up sugar at an even faster rate than the surrounding tissues, even the brain. We call this disproportionate thirst for sugar the Warburg effect, cancer's sweet tooth. And in the field of medicine, we've actually learned to capitalize on that for diagnostic purposes. Dr. Joanna Fowler, who I've actually had the privilege of working for at one point, discovered with a team of scientists a new way to synthesize a radio-labeled glucose molecule that can be injected into patients. There's a radioactive fluorine inserted in the second position. It's called 2-fluorodeoxyglucose. And what this FDG molecule does is you inject it into a patient and it will rapidly partition into tumors and cause them to illuminate under a non-invasive imaging technique called positron emission tomography, or a PET scan for short. And you can see an image of that over here, taken from Wikipedia, and this rotating GIF. This person was injected with FDG, and understandably, the brain is going to glow quite a bit because it's a very sugar-hungry tissue. But oh no, in the liver, we have metastatic cancer, which is glowing because it is taking so much of this radioactive, shining sugar molecule. Dr. Fowler and the team that she worked with, they're all geniuses and heroes. And it's truly remarkable how FDG has just saved countless lives. And that's why I usually get very irate when people say, oh, scientists have the cure for cancer, we just don't tell anyone. Well, believe me, we want to solve these problems. It's just really difficult. But when we make breakthroughs, it's certainly worth it. Now, before we put glycolysis to rest, at least for the time being, we need to mention its cousin, gluconeogenesis. Our bodies run on sugar. We can also make it too. And that's important because our brain demands 120 grams of glucose every day to keep the lights on. For the entire human body, it varies person to person, but on average, it's going to take about 160 grams. Now, diet and glycogen alone barely meet this quota. This is going to be a problem if you ever skip a meal or if you did a little bit of cardio that day, which is going to blow through your sugar and glycogen stores. So the liver needs a way to convert pyruvate back into glucose under fasting conditions so we can maintain this brain activity at all costs. Now, you might think that gluconeogenesis is simply the opposite of glycolysis, but that's not true. Even though 7 of the 10 steps are catalyzed by the same enzymes, there are key difference points. We call these bypasses or choke points. These are going to be regulatory steps that will help us toggle when the cell is to enter the glycolytic mode or the gluconeogenic mode. These difference or choke points at step 1, step 3, and step 10 of glycolysis have to be bypassed to ensure that the gluconeogenic pathway is also going to be spontaneous, thermodynamically favorable, exergonic. Think of it this way. If gluconeogenesis were truly the reverse of glycolysis, it would be a very endothermic process, but that's not true. Both pathways are going to be exergonic, negative delta G. How do we ensure that both processes are spontaneous? Well, the gatekeepers, the regulatory steps of glycolysis, indeed all have very negative delta Gs. That's why overall it's so spontaneous. But the gluconeogenic alternatives of these processes also have very negative delta Gs. And the enzymes that replace these glycolytic gatekeepers are pyruvate carboxylase and PEB carboxykinase. Both of these enzymes are going to replace step 10 of glycolysis. To replace the third step, the PFK gatekeeper of glycolysis, we are going to use F16BPase. And step 1, the last gatekeeper we have to bypass, is going to be catalyzed by glucose 6-phosphatase. So there you have it. These are the gluconeogenesis gatekeepers. And these are the glycolysis gatekeepers. All the other enzymes have insignificant, more or less, delta Gs, and therefore are not really regulatory steps. So I've listed them in the table over here. Let's go through these new gluconeogenic steps one at a time, because these are the only parts that are truly different between glycolysis and gluconeogenesis. The first bypass reaction in gluconeogenesis is again the most complicated because it's actually two different enzymes working in succession, but let's just begin with the first one, pyruvate carboxylase. This enzyme's job is to install a CO2 molecule onto pyruvate, which is going to prime it for reactivation by essentially plugging on a good leaving group. And we can see that in the reaction equation over here. We have a 3-carbon pyruvate sugar acid plus a 1-carbon bicarbonate ion. This is what you get when you dissolve carbon dioxide in water. We need a little bit of ATP to coax these two molecules together to form the 4-carbon sugar acid oxaloacetate, and then ADP and PI are, of course, the byproducts. So how does this work? Well, the enzyme, any kind of carboxylase is really interesting because they're going to employ this biotin cofactor, shown over here. And that biotin cofactor is going to be constantly hopping between two different active sites, site 1 and site 2. In the first site, what we're going to first do is combine our bicarbonate ion with an equivalent of ATP to form carboxyphosphate. Carboxyphosphate is activated CO2. Now that it's activated, our nucleophilic biotin can attack the electrophilic carboxyphosphate, and it's going to substitute in the place of that phosphate. It's going to kick it out, and it's going to grab on and latch to that CO2 group, shown over there, as now a carbamoyl moiety. So site 1, the goal is activate the bicarbonate for biotin carbamoylation.
we now have attached a COO onto biotin, which is going to swing on over like a lasso to the second active site, where we're going to take that CO2 and slam it onto another molecule, stick it on. And we are going to specifically stick it on to pyruvate, as described. By doing an aldol condensation, we can add this 3-carbon unit to this 1-carbon unit to get the 4-carbon oxaloacetate. This CO2 makes the next step possible. And since this is the first reaction in the first bypass of the gluconeogenesis gatekeeper reaction, it's really important that we tightly regulate it. Pyruvate carboxylase is practically inactive in the absence of acetyl-CoA. It is a very powerful allosteric activator. And that makes a lot of sense because acetyl-CoA is going to accumulate in tissues that have plenty of carbon to use, and there's not a lot of drive to burn it for ATP. So let's take that acetyl-CoA and maybe go back to a glucose molecule. The allosteric inhibitor for this process is ADP, which is a partially depleted battery. If there's lots of ADP in the cell, that means we're using lots of ATP and probably need a little bit more. So maybe it's best if we took the pyruvate and sent it to the furnace after all. We can shut down this gatekeeper by using ADP accumulation to our advantage. I'll briefly remind you that the first bypass to gluconeogenesis is a 2-step process, and this enzyme catalyzes the second half, and that's PEP carboxykinase, which is often called PEPCK for short. So what does PEPCK do? Well, it's going to drive off the CO2 group that we just included within our oxaloacetate molecule. We're going to decarboxylate it, all right? And what gives? Why are we driving off the CO2 unit that we just put on? Didn't we just invest an ATP, a precious ATP, into making our oxaloacetate? Well, we're doing that because when we break off the CO2 unit that we just installed, that is going to let us move these electrons over and form a double bond over there. And that's going to prompt this CO group in the middle to become phosphorylated with the help of GTP, which you can kind of think of as ATP. It's also a source of energy and a phosphoryl donor. So the product that we get from this PEPCK reaction, we go from a 4-carbon substrate back to a 3-carbon one, and it's specifically going to be something with a double bond next to a phosphate group, a phosphoenol, specifically phosphoenol pyruvate. Doesn't that sound familiar? All this effort, and what we got was essentially the 9th intermediate in the glycolytic pathway. Perhaps it's becoming more clear to you now that the reason we put CO2 into our pyruvate molecule was to create an entropic driving force to the reaction. What does that mean? When you generate gas, you're going to create a lot of disorder. And creating disorder is very thermodynamically favorable. We took this 2-step detour, this long way back to making our PEP because in doing so, we're able to ensure that the delta G of the reaction is going to be very negative, negative 25 kilojoules per mole. That's what makes these two successive reactions the first gatekeeper. They're going to be irreversible and help commit those substrates to gluconeogenesis. Now, we want to be sure about committing these valuable molecules to the gluconeogenic pathway, so it's also going to be regulated, in this case, negatively by ADP, which is similar to pyruvate carboxylase. And something else I want to bring to your attention is that we really need to be certain we're moving forward here because this reaction needs to occur twice per glucose. All right? Glucose is a 6-carbon compound. Phosphoenol pyruvate is a 3-carbon one. So we have to do this again and eventually merge those two phosphotriose sugars back together. So this entire reaction of pyruvate plus ATP plus GTP plus bicarbonate needs to be multiplied by 2. So already, this process is looking rather expensive. The second and third bypass reactions to gluconeogenesis are way easier to comprehend, in my opinion. They are specifically FBPase1 and G6Pase1, and both of them have a common goal of removing phosphates, specifically the phosphates that we installed through that set of first bypass reactions. Now, in terms of their mechanism, we're not going to go through it step by step because it's pretty simple and they have an identical mechanism basically. It's a phosphate hydrolase. They're going to activate a water molecule, which is going to then attack the phosphates one at a time and kick them off through a nucleophilic substitution process. Now let's talk a little bit more about the specific role fructose 1,6-bisphosphatase does. Its job specifically is to decommit sugar from energy metabolism. All right? You can think of this as the spiritual reverse of PFK, phosphofructokinase, and its job was to commit sugar to energy metabolism. So what fructose 1,6-bisphosphatase does, we have our fructose 1,6-bisphosphate. It has phosphate on the 1 position, phosphate on the 6 position, and it's going to take that water molecule and it's going to substitute out and cleave off the phosphate in the 1 position, giving us fructose 6-phosphate as the product. In terms of its regulation, you may notice that it has some common effectors between it and its cousin, phosphofructokinase. However, their effects on the enzyme are reversed. With PFK, citrate slows it down. Here, when citrate accumulates, it speeds it up. That's why this and its complementary enzyme PFK are choke points. You activate one, you deactivate the other. And so that's a really handy way of tuning 2 different metabolic pathways with one flip of the switch. Conversely, you may remember that F26BP and AMP were very potent activators of PFK. Here, for fructose 1,6-bisphosphatase, they're very potent deactivators. And that makes sense because when those two substrates accumulate, that means we have a very high need for ATP. Let's not waste it recreating glucose. Let's just burn it and try to get some more energy. Now let's swing on over to our last bypass reaction catalyzed by glucose 6-phosphatase, and its job is to decommit sugar from the liver. We are going to take that last phosphate lingering on the 6 position, and we are going to chop it off, bringing us back to a regular old glucose. If you recall, the first step of glycolysis was to commit the sugar to the cell. And here we see what I like to call the spiritual reverse of it. We are now decommitting it from the cell. We are free to release this glucose into the bloodstream so it can swim on down and feed the tissues that desperately need it. And again, you may notice that both of these reactions have large negative delta Gs, 16.3 and 13.8 negative kilojoules per mole, enforcing their roles as gatekeepers, regulatory steps for the gluconeogenesis pathway. Some time back, I said that gluconeogenesis isn't the mere reverse of glycolysis. The following equations should help nail that in. Glycolysis, if you remember, is a 10-step process. It's highly exergonic with a reaction delta G of about negative 63 kilojoules per mole, and is therefore irreversible, thermodynamically favorable. In terms of its energetic products, it's going to yield 2 ATP molecules for immediate use, which is rather nice. Now let's compare all of that to gluconeogenesis. Gluconeogenesis, you may remember, its first bypass reaction actually features 2 enzymes working in succession. So we actually need 11 steps to complete it, not 10. The reaction delta G is also different from glycolysis. It's still rather irreversible with negative 16 kilojoules per mole, but it's not as negative as glycolysis. So these 2 facts are a big flashing red sign that glycolysis and gluconeogenesis are spiritual opposites, but not literal opposites. If it was just glycolysis in reverse, it would only take 10 steps to do it, and the reaction delta G would be overwhelmingly endergonic. It would be positive 63 kilojoules per mole, unfeasibly uphill, and certainly not spontaneous. I also want you to pay attention to the reaction equations themselves. Just by inspection, you can see that the reactants and products of glycolysis don't line up with those of gluconeogenesis. Most glaring, to me at least, are the ATP terms. We get 2 ATP molecules net from glycolysis. It does not take only 2 ATP molecules to do gluconeogenesis. Oh, no, no, no. It requires 4 ATP molecules and 2 GTPs to boot. Think about the implications of this. The cost of gluconeogenesis exceeds the profits of glycolysis. If we were to burn that glucose molecule and get 2 ATP out of it, we would still be in the red. Hence, gluconeogenesis is expensive. It's not free food. It's not free glucose. But it's still crucial to our daily function. Let's say you're hungry or you're working out. Yes, your muscles and brain may be highly catabolic, but your liver is going to still be full from your last meal. It's going to be in an anabolic state, and it will have the ability to make this glucose, even though the process is kind of inefficient, but it can then distribute that glucose to your tissues once they blow through your sugar and glycogen stores. All right? So the next time you're in the gym and you don't experience sudden brain death, this is why. I'm going to cap it off here with this nice summary figure that I wanted to share with you. It contains all of the chemical logics of every step in the pathway for glycolysis. So I figured you could use this to help you study while we prepare for the next topic, which is going to be the regulation of glycolysis and gluconeogenesis. So until then.